The next part of the nephron is the proximal convoluted tubule past the Bowman's capsule. It's composed of simple cuboidal epithelium, and it has many microvilli on the apical surface, as well as extensive foldings of the basal surface plasma membrane. It contains a large number of mitochondria, which provide energy, as well as the basal folding provide increased surface area for extensive ion movement that occur across these cells. Proximal convoluted tubule is the primary site of reabsorption of sodium, glucose, amino acid, as well as bicarbonate. In this picture here, you can see that you have the Bowman's capsule here, the vascular pole, the urinary pole, and then the proximal convoluted tubule in a longitudinal section, as well in a cross section here. One thing to note, and you will definitely be learning this in histology lab, is that if you look at the lumen of the proximal convoluted tubule, it appears to be fuzzy. And it's fuzzy because of the microvilli which are projecting from these cells into the lumen. If you were to then look at the EM of proximal convoluted tubule cell, and you will see that, you will be looking at that in the lab, you will notice a lot of microvilli here on the apical surface, a lot of foldings here on the basal surface, and the cell sitting on the basal lamina here, as well as internally a number of mitochondria can be observed in the cytoplasm. So once the urine travels through the proximal convoluted tubule, the next thing, next place it comes to is referred to as loop of HENLE. Loop of HENLE, I will show you a picture here to make it a little bit clear to understand, is the start after the proximal convoluted tubule, a straight line which is going down and then going up into the second convoluted portion of the nephron here of the tubule. So these straight portions are referred to as loop of HENLE, and here you have thick descending, thick ascending, as well as thin descending and thin ascending portions of this loop. So the thick descending part of the loop is a simple cuboidal epithelium which has reduced microvilli and fewer basal surface specialization than the proximal convoluted tubule. Similarly, the thick ascending is simple cuboidal, similar in appearance to the thick portion. In this picture here, you can see the thick portion. We don't really know whether it's a descending or ascending in this particular section, but they are the thick portions here, quite easily identifiable. So correspondingly, you also have then the thin parts. And here you have the thin either descending or ascending, but the epithelium is a simple squamous epithelium, which does vary depending on which type of nephron you have, and we'll see that in a minute, whether the nephron sits in the cortex or projects into the medulla. Either way, loop of HENLE functions in the regulation of osmolarity of urine. While we are on this picture, I just want to point out that it is a rather complex picture. One thing that you will notice is that you have what appears to be the thick parts and the thin parts, but then again you also have other features which look like thin part of the loop of HENLE, but then you look at the lumen, you notice that it has other features which you would identify them as red blood cells. So this is actually vasculature. So this is vasa recta, and we will see that a little bit later. Here are those two different types of nephrons that we were talking about. So depending on whether the nephron stays within the cortex portion, so it does not penetrate into the medulla, or it's a long loop nephron which passes through into the medulla. Histologically, it's not an important feature. You would not be expected to identify these different types of loops in histology. So once the urine has gone past the loop of Henle, it arrives at the distal convoluted tubule, which is a shorter version of the proximal convoluted tubule, about 1/3 of the length. It has simple cuboidal epithelium, just like the proximal part of it, but the microvilli here are much reduced in size and number compared to the proximal part. Distal convoluted tubule does contain a lot of mitochondria as well as basal membrane foldings for increasing surface area and providing energy for sodium bicarbonate ammonium transport across the cells. So here, same picture again, but in this case, we are pointing to what are the distal convoluted tubules. You compare these with this one here or this one here, you'll notice that the proximal convoluted tubules here are thicker, as well as the lumen is fuzzy, whereas lumen is clearer here in the distal convoluted tubule portions. And again, you see a vasculature here with red blood cells. Here's a picture at the EM level of distal convoluted tubule cell. Big nucleus here, the apical side, the basal side sitting on the basal lamina with infoldings, as well as lots of mitochondria here. Noticeable difference here is this very insignificant presence of microvilli on the apical aspect. The other thing that you would be expected to know a bit about in histology as well as physiology is a feature referred to as macula densa. Macula densa is part of the distal convoluted tubule. This is the portion where distal convoluted tubule comes back to the Bowman's capsule and makes a connection right adjacent to the Bowman's capsule here, and there's an aggregation of these cells or density of these cells, macula densa. And these cells are involved in regulating renal function in two ways. Either they increase the fluid flow when they detect sodium chloride, and then they trigger paracrine stimulation of the afferent arterial constriction, which will be right around here, or they decrease the flow by triggering stimulation of the renin excretion by the adjacent cells which are present in the locality here, and in the increase in blood pressure. So after the urine has gone through the distal convoluted tubule, it makes a connection with the collecting ducts, which you have learned before, but they're clearly visible here as simple cuboidal epithelium. They do have small microvilli and a primary cilium. Primary cilium is pretty much involved in looking at the pressure within the tubule. The epithelium, which is generally cuboidal, can become columnar in nature as the ducts become larger and larger. Eventually, they cluster together, these ducts cluster together in the cortex, in the area which we term the medullary ray, and then subsequent to that, they continue into the medulla. So just to help you understand this portion here, this picture you have seen before, we refer to these portions here as medullary rays here, and then they would eventually run into the medulla and then release their content, the urine, into the calyces here, and then that would eventually end up in the renal pelvis and then out. Just to help you a little bit further, you can see this a little clearer in this histological view. These medullary rays in the cortex, and then these ducts end up in the medulla over here. So the capsule is in this portion here. So when the ducts merge together, they become larger and larger, and they end up at the tips of these pyramids we have seen before, and the area referred to as the papilla, and then they be referred to as the papillary ducts. So as with other kidney tubules, the epithelial cells are active in ion transport, and they move that to adjust the concentration as well as the pH of urine.